Hello everybody, thank you for coming and thank you for the invitation to participate in this session on the next agriculture transformation in the framework of this digital conference on Green Smart Development and Vision 2021. In the next few minutes, I will share the presentation Biotechnology for Agriculture Development, Current and Future Tools, in which I will present some ideas of how an important number of Latin American countries are already considering biotechnology as the key tool for agriculture development. I would like to start mentioning that a few days ago, the world population was more than 7.8 billion inhabitants, and each person on the planet has certain needs and requirements for which agriculture plays a fundamental role. For instance, to supply the food needs of the population and the non-food requirements represented here in raw materials for various industries, fibers, active ingredients for medicines, and even biofuels, this generates both positive and negative impacts on society and the environment. And agriculture must produce and respond to these growing demands in compliance with the Sustainable Development Goals in an scenario marked by the uncertainty of a pandemic that, among other things, is overshadowing other issues. It is under this scenario that science, technology, and innovation are seen as vital for human survival. However, although science, technology, and innovation are important, they are not sufficient, and at least another element must be taken into consideration: the institutionality. Institutionality understood as both institutions and instruments that, at the end, make the things functioning. So, agriculture transformation will require a closer and stronger interaction between science, technology, innovation, and institutionality in order to respond in an efficient manner to society's growing demands. In this context, biotechnology, defined by the Convention of Biological Diversity as any technological application that uses biological systems and living organisms or their derivatives for the creation or modification of products or processes for specific uses, has become, without exaggeration, one of the most important strategic tools for the development of humanity. So, under this broad definition, many activities can be considered biotechnology. Here, I would like to show some of the techniques that brought biotechnology to agriculture. Several of these techniques, although more than 50 years old, are still useful and will continue being used for different purposes. Unfortunately, we don't have time to show how each of these techniques are clearly contributing to the development of future agriculture. And although I will focus on genetic modification today, I want to reinforce the point that biotechnology is much more than transgenesis or genetic modified organisms, or in short, GMOs. Before pass to the next slide, I want to bring a reflection. Agriculture is one of the few areas in which traditional, conventional, and cutting-edge knowledge are coexisting, and the new knowledge does not replace the previous one. And this characteristic will continue being exploited for future agriculture development. Therefore, as a summary of this introduction, for several Latin American countries, the transformation to a futures agriculture started several years ago, thanks to introduction of several biotechnology techniques. As I mentioned before, due to lack of time for this talk, I will focus only on plant biotechnology and specifically on gene modification through transgenesis and genome editing. First, the term genetic modification groups together several techniques that make possible to manipulate the DNA. Such manipulation can involve the introduction, disappearance, or replacement of segments of variable length of genetic information. In addition, for several years now, it has been even possible to edit genetic information in a very precise way. In nature, genetic modification is known as mutation, a widespread phenomenon that has generated the biodiversity on the planet in the form we see today. However, genetic modification is nowadays a process that can be carried out in the laboratory. Genetic modification can be done because there are two facts. The first one is that all living beings contain genes, and the total number of genes in an individual is what we know as the genome. And all genomes have some degree of similarity. For example, the corn genome is around 60% similar to that of humans. The chimpanzee genome is around 93% similar, and among humans, our genome is 99.9% similar or identical. This means that only 0.1% of our genome explains the diversity we see in the more than 7.8 billion humans. Thus, from the genomic point of view, the world is analogous to a large library, where the complete information of each individual is written in a book called genome, and the pages of each book are the genes. In the case of humans, the genome book contains about 20,000 pages, while for a virus like COVID-19, the book has no more than 10 pages. Now, the important thing about genes is that they contain the instructions to generate a product, usually a protein. And this applies to all living organisms, whether it's a plant, an animal, or a microorganism. Just as the gene is analogous to a page of a genome book, DNA is the language in which the book is written. And with this, we round off the idea that living beings contain genes. The second fact that makes genetic modification plausible is that it is possible to isolate and manipulate these genes and genomes. This is done by means of molecular biology tools that allow identifying, isolating, and copying that gene, that page of that book. And it is possible by means of molecular biology tools to introduce that page or gene into the genome or another organism. This is what is known as transgenesis. But biotechnology is evolving very fast, and now genetic modification has a very powerful technology that makes possible to literally edit a single letter of the page of the book called genome. Although there are different techniques for gene editing, the most famous is known as CRISPR-Cas. And it is so important because it is potential application that even though was invented and reported only 8 years ago, the researchers who developed it have been awarded with the Nobel Prize in Chemistry last year. Just to emphasize that gene editing is not transgenesis. Now we can see more in detail the use of genetic modified technology in agriculture. In short, transgenesis is a technology that allows the introduction of genes that have been identified, isolated, and copied from an organism of one species into another species without the need for sexual reproduction. In this case, we see how a conventional corn that is affected by an insect attack can be modified by including a gene extracted from a bacterium, in this case, Bacillus thuringiensis. That produces a toxin for the insect, resulting in a corn that is tolerant to the insect attack because the instructions to the genome of the new corn allow it to generate a protein that is toxic only for the insect, but not to other individuals. This characteristic of insect resistance brings additional benefits since the corn does not suffer damage and consequently is not affected by fungi, and it does not need additional insecticides. As an indirect result, it generates a higher production. For its quality, the product is having a higher price, and it generates a higher profit for the producer. Currently, there are events with insect or herbicide resistance or with both characteristics expressed simultaneously in corn plants. To return to our analogy, it is as if 2 pages have been added to a 60,000-page book. In addition, these characteristics, together with the economic benefit, make farmers continue using this genome modification or genetic modified seed. Here is a real example of a Honduran farmer who, with almost a 4% increase in his production cost, receives up to 62% more profitability by using genetic modified corn. In this slide, I present a summary of genetic modified crops during year 2019, with more than 990 million hectares growing in 29 countries and linking around 17 million farmers, of which more than 90% are small-scale farmers. Important to mention that thanks to the technology, this technology is being managed with strict protocols that guarantee the safe and the innocuity of the process. And the discipline behind this is called biosafety. Noteworthy to mention that parallel to transgenic agriculture, organic agriculture is increasing as well, which shows us that all types of agriculture are growing because at the end, they have the same objectives, to produce more and to produce better, through different routes, but the same objectives. Continuing with this fact traveling on genetic modification, we need to talk about genome editing. And for that, I would like to begin with focusing on a few facts. Over the last 10 years, there have been remarkable advances in biotechnology. Several techniques have been generated and have given shape to what is known with different names. For example, genome editing, gene editing, new breeding techniques, precision breeding techniques, plant breeding innovation, or as we call in IICA, precision biotechnology. And this is important because it shows us how difficult it can be to regulate the same thing with so many names. Another fact, precision biotechnology uses advanced molecular biology techniques, for example, zinc nuclease finger, TALEN, CRISPR-Cas, that facilitate specific, precise, and efficient modifications of a locus in the genome. And a fact of enormous relevance, all these techniques allow genome manipulation, but they are different from the technique of genetic modification based on transgenesis. However, the very high precision of these techniques contrast with the complexity of explaining them in detail, which constitutes a challenge in terms of communication and public perception. Unfortunately, we don't have time to explain in detail genome editing techniques. Therefore, I just want to mention that the genome editing methodologies developed so far make use of the enzymatic DNA breaking and the DNA repair capabilities. At present, by means of one of 3 techniques, zinc finger nucleases, TALENs, or CRISPR, it is possible to make breaks at specific sites of the double strand of DNA. These breaks are made with specific nucleases, known as SDN, for site-directed nucleases, which recognize specific sequences. Then, the repair can be done by 2 mechanisms, the non-homologous enjoining and the template-assisted repair. These are 2 natural existing DNA repair mechanisms. The precision of biotechnological techniques is indispensable because genome association studies have shown that single base changes are usually responsible for variation in the characteristics of elite cultivars. And by making small changes in a very precise manner, there are much lower risks than those associated with LMOs that have risks associated or identical to, for example, the risks associated with conventional or organic crops. This is supported by these 3 facts. First, gene editing alters only a few nucleotides. Second, the changes are very likely to be found in nature. And third, once the edited product is generated, there is no way to distinguish between the change due to natural mutation or the change due to gene editing. Another fact is that genome editing, in particular CRISPR-Cas, have received much publicity for their potential applications and impacts. For example, in medicine, environment, agriculture, livestock, ethics, legal, economics, etc. Unfortunately, some groups have equated gene-edited products with LMOs. And in fact, some of these groups have stated, this is just another name for transgenics. And GMOs, by another name, are still GMOs. Moreover, here it is time to reiterate that gene-edited products are not necessarily transgenic ones. From a regulatory point of view, the current questions associated with gene editing include what to regulate. Would the Cartagena Protocol apply? Would it not apply? What there is a consensus on is that there is no need of a new category of products, and that what is needed is a case-by-case evaluation, inquiring about the transgenic nature of such a product. On this basis, the regulatory frameworks of several countries establish that if a gene-edited product falls under the Cartagena's protocol definition of LMO, it will take the LMO regulatory pathway. But if the edited product does not meet the LMO definition, then it is treated as a conventional product. In the Western Hemisphere, there are several countries with regulation for gene editing. In addition, there are regional resolutions that support the safe use of the technology. For example, resolutions of the Southern Agricultural Council, CAS, and the Custom Union between Guatemala, Honduras, and El Salvador. The important thing is that they all promote the safe development of the technology. We don't have time to talk about each country's regulations, but I just want to point out that the Western Hemisphere is leading the regulation, the discussion, and the use of this technology. In contrast, in the European Union, the Court of Justice considered that gene editing should be regulated under the law restricting the use of LMOs. And recently, the result of a study on the status of new breeding techniques and the European law has been presented, and eventually a new decision will come out. And close to the end of this presentation, I would like to tell you that because of the importance of biotechnology and biosafety for agriculture, IICA has been actively working on 3 types of actions. First, by providing technical support to the national institutions, for example, by reviewing policies and norms, proposing adjustments to norms, and to support countries for the preparation for participating in international fora, such as the COP-MOP of the Convention of Biological Diversity, or the Cartagena's Protocol on Biosafety, as well as for other fora like OECD or WTO, between others. The second group of actions focus on providing training in specific issues, for instance, in biosafety, on topics such as risk assessment, synthetic biology, and gene editing. And the third group of actions are related to organizing or co-organizing exercises of efficient communication of biotechnology. In terms of supporting institutionality, you can see here several examples of national, binational, and regional initiatives, some of them involving GMOs, others related to genome editing, and even initiatives regarding biological inputs for agriculture, which is quite relevant for organic agriculture and agroecology. Because here there is an important message. Biotechnology is a basis for all forms of agriculture. In terms of training, education, and communication, you can see some examples. Here, details of a seminar on risk assessment in which we were able to mobilize 42 regulators from 20 countries to visit the ponds containing genetic modified salmon. More recently, in year 2018, details of the seminar on gene editing for hemispheric regulators that was held in Cali, Colombia. Noteworthy to mention that from this seminar, it was proposed the basis of the International Declaration on Agricultural Application for Precision Biotechnology, which was presented to the Committee on Sanitary and Phytosanitary Measures of the WTO. And in February last year, we were able to conduct a practical training in gene editing in a joint activity between IICA and the Alliance Biodiversity CIAT, again in Colombia. And regarding communication, we have carried out several activities that are shown here, mainly virtual activities due to the pressure of the pandemic, the COVID pandemics. To conclude, some remarks that reinforce the idea that future's agriculture in the Western Hemisphere looks very promising. The first one, biotechnology have developed several tools that can be used and are already being used by farmers. The second point, regulatory frameworks promote the safe use of the technology, and a growing number of Latin American countries either have developed or are implementing them. Third, precision biotechnology allowed overpassing certain technical limitations from other technologies, and gene-edited products for agriculture are already in the pipeline, and in few years, they will be available for being used by farmers, all-scale farmers. Fourth, regulatory cooperation is a key element for closing gaps, and Latin American region is working very hard for strengthening regulatory relationships. And five, training and communication must be reinforced to different groups. It is clear that many efforts for this purpose have been put in place in the past. However, more work on communication is required on a regular basis because technologies will continue to appear and communication must be next to them for the proper understanding of producers and consumers. Biotechnology has demonstrated that can contribute to the green smart development of agriculture by providing opportunities to all types of producers and production systems. So, let the producer to choose the technologies to use, because future's agriculture is today. Thank you very much for your attention.